All right, guys, to spend a little time looking at 6.2 and 6.3, what we covered in class today, just a recap for those of you that want to go back to the information that we covered today. So again, objectives. In order to be successful in the AP exam, you need to meet the following three objectives: understand the different types of RNA, explain the three-step process of transcription, and explain the modification of RNA after it's been transcribed. And that gets us all ready for translation. So this is just transcription. Okay, just an overview. Remember, transcription is synthesizing RNA from DNA, okay? Which is this first picture here, okay? DNA to RNA. Translation is going from RNA here to protein, okay? So this is, if we were to split this picture in half, here, transcription and translation. How do you tell the difference between these two? Because they're both T words and you mix them up really easy, right? When we think of language, we're going from someone talking, okay? And a scribe often had the job of writing it down, okay, on a piece of paper. Now, it's still the same language. It's still written. It's still words, right? Okay, in the same language. Say we're doing this in like English, all right? And then if we're translating a language from English to Spanish, okay, it's a completely different language. Okay, we're using completely different material here, and that's what happens when we make a protein. Is we're going from we have nucleobases, and then we use amino acids. Okay, so there's a completely different biomolecule being used here. That's how you can tell the difference. Okay, how is mRNA read? Okay, we're using the triplet code. This is something to keep in mind as we continue. All right, these are called codons. All right, codons. They're three nucleobases that are the genetic instructions for how to write a polypeptide. So the way that codons are read is they're read in threes, and they code for a particular amino acid. Okay, so that's important to keep in mind, is that they're written in threes. These three words that code for one amino acid, okay? Something to keep in mind also, in RNA, uracil is going to replace the T. Okay, so you can see here, A is going to pair with U. Instead of, if it was a piece of DNA, it would pair up with T. But here, when A is matched up with RNA, it is uracil. We have three types of RNA, messenger RNA, ribosomal RNA, and transfer RNA. How are they different? Messenger RNA is like the top dog. They're going to carry information from DNA in the nucleus to ribosomes in the cytoplasm. They're doing most of the work. This is the most common form of RNA, is mRNA. Ribosomal RNA is going to combine with protein to form the ribosome. So think of ribosomal RNA as part of the structure of the ribosome that bring tRNA and mRNA together. Okay, whereas tRNA, you can see it's kind of shaped like a T. It's going to transfer our amino acids to the ribosome to build proteins. It's going to hook on to our mRNA here, our codon with its anticodon, to build a polypeptide chain. Okay, we're going to focus more on mRNA to begin with. In eukaryotic cells, we go from DNA to pre-mRNA. mRNA is going to be processed into mRNA. It's going to leave the nucleus through these nuclear pores, and then we're going to undergo translation. So we'll talk about this process here in eukaryotic cells. Okay, transcription, we have one key player. This is RNA polymerase. It's going to rip apart our DNA, hook RNA nucleotides together from the DNA code, and then it's going to spit out RNA on the other side. Okay, so here's our RNA polymerase down here. Just as a quick overview of the three regions to know about DNA, during transcription, you'll hear three regions talked about. Okay, we have our promoter region, we have our terminator region, and our transcription unit, or a transcription region. All right, we're going to talk about these in depth in a second. You also need to know the transcription steps. Okay, so you need to know three regions of DNA, three transcription steps. They all correlate together. The initiation step is when we have our transcription factors. Okay, so imagine the RNA polymerase has like 3 other proteins as part of it, okay? They're floating around in the cell. And they're going to recognize our promoter. Okay, this is our promoter region. That's what I just mentioned earlier. And there's a really small sequence on our promoter region right here that is going to have, if I were to zoom in it, it would have this, this code, or this set of nucleic bases. Okay, and this is these are called our data box. I'm gonna get rid of our data box, sorry. There we go. So we have our data box. Our data box is recognized by our RNA polymerase and other transcription factors that are all going to come together, and they're going to hook on to our promoter region. Okay, they're going to open up the DNA that you can see here. It's been opened up here. Okay, and we're going, so say this is our RNA polymerase. It hooks on, and it's going to start to travel along our DNA. Okay, now it's going to hook on to a 3' end. Okay, so this is 3', and it's going to, this part here is 5'. So it's reading from 3 to 5. Okay, as it's going along during the elongation phase, continuing to unwind the DNA, it's going to spit out RNA 5' to 3'. So it's hooking on RNA pieces on the 3' side, constructing it 5' to 3'. Really important to know that, okay? Then termination, this is the part where our RNA polymerase reaches a terminator sequence. So that's that green region here. It says stop, basically. Okay, and this terminator sequence is going to tell our RNA polymerase and the other transcription factors to dissociate, to leave, and then our RNA is going to be spit out, and it's going to undergo further processing in the nucleus, and then it's going to end up leaving the nucleus through the nuclear pores and go out into the cytoplasm, okay? Now, initiation, I talked about that kind of in depth already. Remember that we have our promoter region on our DNA. It recognizes our data box. RNA polymerase is going to hook on, and it's going to start to unwind our DNA strand. Okay, so this initiates RNA synthesis. When it's unwinding it, it recognizes that data box, hooks on, and then it starts to make this RNA transcript. Okay, that's going to be our initiation sequence. Remember that we're only translating transcribing only the template strand. Remember, this other strand is just kind of hanging out up there. It has nothing to do with RNA polymerase. It's just kind of hanging out. During elongation, our polymerase, like I said, is going to move down the downstream. And as it travels downstream, it's going to unwind our DNA and start to tack on this 3' end, hooking on little pieces of RNA. Okay, that it'll tack on this 3' end. Okay, this will carry on until it reaches the termination process. But before we get there, if you're having trouble on this video, helps to explain directionality. So remember, we read 3 to 5. And you write down 5 to 3. So it's kind of, does a good job of explaining, if you want to go back to the slides, of directionality of DNA and RNA. So then we have our termination. Okay, this is when we've hit our termination region on our DNA. The RNA polymerase is going to detach, as well as the other transcription factors. And then we'll have our completed RNA transcript ready to carry out further processing. Okay, another picture to visualize kind of how the bases are pairing up. Take note of which ones are pairing up. C's and G's are always pairing up. A's and T's always pair up here. Okay, note that we don't have on this side we have T's. On the DNA side, on the RNAs, they are U's. Okay, this is important to know when you're going to be practicing transcription on your own. And then if you want to watch this animation on your own, it's really good too, just to kind of visualize what's happening. It doesn't go quite as in-depth, but it's good to visualize the process. And then when we're almost done with this process, we have our mRNA modification. So mRNA needs to be matured a little bit, and it does that in 3 ways. First, we're going to put a 5' cap on the end here, okay, which is just a modified guanine with 3 phosphates on the end of it. And basically this is for it to be recognized by ribosomes, as well as added protection from hydrolytic enzymes. Second, we have 3' tail at the end, this poly A tail. This is lots of of adenines thrown on the end. And this is mainly protection, because think of like Pac-Man, okay? Of these hydrolytic enzymes is Pac-Man that are just going to be kind of chomping away, eating out these A's. And it's to help prevent the actual message from being destroyed. And so it helps to protect also recognition of the ribosome, word end translation, and for transport of the mRNA. Okay, so I kind of listed those here, and if you have questions on that, feel free to ask. And then our third mRNA modification is RNA splicing. Okay, all right, and this splicing causes exons, which is our non-coding or our coding region that actually has the code for proteins, to be kept, our introns to be kicked off, to be cut out by something called the spliceosome, which is going to hook on here. The intron is going to form a lasso, it's going to cut it, and then we are left with our spliced mRNA, which is our mature mRNA. Now, the video referenced alternative splicing and regular splicing. So alternative splicing just means that we can, say we cut out this exon, and we only have the 5' exon and the 3' exon together. That'll make an even shorter protein. So this is where we get different varieties of protein, depending on if we can alternatively splice it a different way. You get 2 different modified proteins. So you have just excision of introns results is called splicing. We can also have alternative splicing depending on which exons you're actually excising. Here's a picture to visualize, for you to help visualize kind of the whole thing that we're doing here. So this is where we stopped. Now we're going to talk about translation. Okay, translation, you need to understand the 3 roles of the types of DNA or RNA. So we talked about the 3 types. We're going to look at the 3 roles that they play. You need to explain the 3-step process of translation and learn how to read the codon code for amino acids. The roles that each of these RNAs are going to play is what we're going to talk about first. So mRNA is going to be the message, okay? It's coming from the nucleus. It's going to be read along its codons by tRNA's anticodons at the ribosome. All right, and so when we have our tRNA attaching to our mRNA, it contains what's called the anticodon, which is the opposite of the message that the mRNA has, the nucleotide differences, okay? And this is also going to have an amino acid tacked on at the top, depending on what the anticodon codes for. And we'll talk about that in a little bit when we learn to read when we learn to read an amino acid or a codon chart. Okay, RNA, just think of the ribosome. Okay, it's part of that ribosome subunit. Okay, but part of both of these subunits. So imagine it's kind of intertwined in here. This is the site of where our tRNA and our mRNA are going to come together. The site of mRNA codon and anticodon coupling. It's going to these are going to couple at 3 different parts. So the A site is where our tRNA is going to come in, and it's going to have carrying that growing polypeptide chain. Okay, so it's going to have a polypeptide chain here. It's going to move over to the P site. Okay, our tRNA is going to have our other tRNA take away the polypeptide from that first one. So the P one here is, as it shuffles along, our P that's now in the E is going to get kicked out. Okay, so again, all of these sites are holding different tRNAs at different phases of polypeptide synthesis. Okay. You can kind of visualize that here. It does a better job of diagramming this process. So here we've got at our P site, look, we've got a tRNA that is next to this tRNA. This second tRNA binds to a matching codon. Okay, you can see in the second picture, this set of amino acids is going to hook on to the amino acid in our A block. Okay. So our new polypeptide is linked to the new amino acid. Here we see them all in our A block. Our P block no longer has an amino acid. The mRNA is going to shift forward here, exposing this codon. Exposing this tRNA. It's going to leave, and then now this new codon is ready for a new tRNA to grab onto it. Okay, Now we have, in translation, we have three processes very similar to transcription: initiation, elongation, and termination. Initiation is where we have our tRNA recognize our mRNA, and they're going to pair up, okay? Then what's going to happen is this is like the signal of like, hey guys, party here at my house, and then it's going to recruit the ribosomal subunit, which is actually several parts. They're going to come together and join the party. During this party, it's going to move along the mRNA, the mRNA is going to go this way, where we recognize different codons with different peptides being formed. New tRNAs are joining, making our peptide chain, okay? And then our tRNAs are getting kicked off. Now, this is going to happen only until we have a set of stop codons. So a 3-letter codon that says stop. There's 3 different sequences that we can have, and we'll talk about that in a second. Once we reach that, once the stop codon reaches that A site here, then it's going to stop. Our polypeptide is going to kick off, and it's going to be a polypeptide chain that's going to undergo further processing called post-translational modification, but you don't need to know that right now. So when we're making polypeptides, when we're undergoing, this is called central dogma, okay? The process of DNA being transcribed into RNA, being translated into protein. Okay, so you should be able to be given a DNA sequence, all right, translate it into RNA, and then further transcribe it into RNA, translate it into protein. How do you take it from RNA to protein? Let's talk about that. Okay, so you have all proteins, or all RNA, remember, is read in these codons, these 3-letter sequences. So let's read AAA, and you can have a variety of types of codon charts. These are the 2 most common ones. This one's a little bit older, so you may have something more new and fancy like this one, but let's look at our first one, AAA. Okay, how do I read this? I'm gonna start in the middle, identify my first codon, A. Identify my second codon, A. Then my third codon, okay, here in this region, we only have one option for A. So what does that code for? Lysine. That means that our tRNA, remember, it kind of looks like something like this. It's got the codons on the bottom. That means that lysine is going to be attached to the top if it has 3 codons that are A. I can't do it. A. All right. UCG. How do we read from the second codon chart? We're going to start with the first letter over here, U. Check. C. Check. See, it says second letter. And then within this box that we identified, we're going to check off the G one. Okay. What does that tell us? It codes for serine. You would just need to know SER. And serine actually has 4 combinations. All right. Now, why doesn't it have 4 combinations here? Plot twist. We actually have 2 series of combinations that can grant us serine. Okay, so here we've got those that we just identified in our block. Here we actually have, if we look at A's, okay, there's 2 combinations that can get serine here. So they tell you the same information, these 2 different codon charts, just you visualize it a different way. I kind of prefer this one, but you won't always have that one given to you during a question. I would like you to take a moment, pause the video, try to figure out what is AUG, what is it going to code for, and why is that important? Okay, AUG is going to code for methionine, and this is our start codon. Okay, this is what always begins protein synthesis. That's the first tRNA that's going to be recognized at the mRNA. And that's going to recruit the ribosomal subunit and then begin the elongation process. Okay, so take a moment, try to fill in this chart. We did this one together in class. And then check your answers based on what you got. Just try your best to fill it in, okay? And then we'll try to fill this one in. Try your best, fill it in, and then I'll go through the answers. So pause the video. All right, welcome back. First, we have a polypeptide chain, okay? This is our protein being made. It's not quite a protein yet because it hasn't been folded. Our polypeptide chain is being kicked out of the ribosome comprised of rRNA, okay? So RNA, ribosome, kind of interchangeable at this point in your understanding of biology. Okay, where do we get this polypeptide chain from? It's being made based off of a particular an anticodon. Okay, it's an anticodon because it's opposite of the codon message on mRNA. Okay, and what happens is we have our anticodon paired up with our codon. It recruits a certain polypeptide to be attached to another one, and the thing that carries the polypeptide is called tRNA. Okay, so go ahead and try to tell that story on your own. At this point, this is what you need to know for transcription and translation. We're going to dig into problems that can occur as a result of poor transcription and translation. So I'm going to have you make sure you watch the AP daily videos for Friday's class, okay? Because we're going to dig in depth again, and I don't want you to get behind, okay? Nice job today. Thanks for putting up with my really fast presenting. Bye.